Good day everyone, this is Group 2 and this is our video presentation. Our topic is predict the effects of hypertonic, isotonic, and hypotonic environments on osmosis in animal cells, and differentiate endocytosis and exocytosis.
I'm GENNYROSE MARIÑAS, and I'm going to discuss the effects on osmosis in animal cells. But first, what is hypertonic? Hypertonic solution is solution that contains more dissolved particles, such as salt and other electrolytes, than is found in normal cells and blood. We all know that solution means a special type of homogeneous mixture composed of two or more substances. So back in the main topic, it said that the effects of hypertonic environments on osmosis in animal cell. But what is osmosis? Osmosis is the swelling up and bursting of cells when made contact with a solution that will cause this. Hypertonic solutions effects on osmosis in animal cells. Hypertonic solutions have less water and more solute such as sugar or salt than a cell. Seawater hypertonic. If you place an animal or a plant cell in hypertonic solution, the cells shrink. Because solutes water, water moves from a higher concentration inside the cell to a lower concentration outside. It means the solution has a higher solute concentration than the cell. A net movement of water molecules out of the cell causes it to shrink. So the water or the H2O is out. It is like balloon, but think that inside the balloon there's a particle or cell. Ay sa animal cell, di ba parang bilog siya? Kinompare ko siya sa balloon kasi yung cell na yun, sa loob nun, nandun yung water, then yung particle. Ang nagpe-present ng animal cell is yung balloon, then yung water is yung hangin. Kapag lumabas yung hangin, yung particle sa loob ng balloon, mababa siya, ang mangyayari, yung hangin is lumalabas. Parang ganun din sa animal cell na lumalabas yung water sa kanya. So, the animal cell may continue to swell until they burst. That, always remember, the cell shrink and water is transported out of the cell. And lastly, solid concentration inside the cell is lower.
Good day everyone, I am Baby Leslie P. CALVARIO and I am here to talk about the effects of isotonic environment on osmosis in animal cells. Next slide please. So let us find out what is an isotonic solution. An isotonic solution is any external solution that has the same solute concentration and water concentration compared to bodily fluids. So this isotonic solution means that no osmosis will occur because same components of the same solution and the cell is the same, so nothing will happen, no reactions will happen. Next slide please. Isotonic solution is a solution in which the concentration of water is the same as that of the cell. So when an animal cell is placed in an isotonic solution, water will neither flow out nor in. No osmosis will occur, so nothing will happen to the animal cell. As I have said earlier, nothing will happen in isotonic solution because same component means that nothing will happen. Examples are, if the solution has hydrogen and oxygen, then the bodily fluid should have the same components as well, like hydrogen and oxygen. Because if none, then this solution is not isotonic. We can see from the picture there that there's hydrogen and oxygen, and inside the cell there's also hydrogen and oxygen. That's all, thank you.
Hello everyone. I will discuss about hypotonic. Let's start in learning what is hypotonic in English dictionary. Hypo means below or what we call less than normal hypotonic. It refers to a solution that contains a lower amount of solute as compared with the solute concentration in the other solution across a semi-permeable membrane. So, hypotonic means having a lower atmospheric pressure that a particular fluid typical body fluid or intracellular fluid. Tap water and pure water are hypotonic. A single animal cell, like a red blood cell, placed in a hypotonic solution will fill up with water and then burst. Imagine, yung egg lalagay sa vinegar. Tapos, manonotice mo yung egg lalaki ng konti because the water in the vinegar can enter the egg through the membrane, moving from the vinegar water concentration in vinegar to the lower concentration in the egg because water is hypotonic. So in short, in hypotonic, the water will get inside the cell. So the mass will also increase and burst when it could not carry the water any longer.
And now we will differentiate the endocytosis and exocytosis.
But before that, what is endocytosis? Did you know endocytosis is the process by which cells takes in substances from outside of the cell by engulfing them in a vesicle? This can include things like nutrients to support the cell or pathogens that immune cells engulf and destroy. Endocytosis is the taking in of matter by a living cell by invagination of its membrane to form a vacuole. It's a cellular process. Endocytosis is in which substances are brought into the cell. The three types of endocytosis are phagocytosis, pinocytosis, and receptor-mediated endocytosis. The first one, phagocytosis, also known as cell eating, is the process by which cells internalize large particles or cells like damaged cells and bacteria. If the material of the cell takes in is particulate such as bacterium or fragment of organic matter, the process is called phagocytosis. And the next one, pinocytosis, also known as cell drinking, is common in plant and animal cells. During pinocytosis, the cell takes in substances from the extracellular fluid that it needs to function. These include things like water and nutrients. So pinocytosis is if the material of the cell takes in is liquid, it is called pinocytosis. And the last one, receptor-mediated endocytosis. As receptor-mediated endocytosis is a process by which cells absorb metabolism, hormones, protein by inward budding of the plasma membrane. Specific molecules such as low-density lipoproteins LDL are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of membrane. In the area of the pit, when appropriate collection of molecules gathers in the coated pit, the pit depends and seals off to form a coated vesicle which carries these molecules into the cell.
Hi, I'm KARLO SARMIENTO, and earlier Simon explained to you what is endocytosis. Now I'm going to explain what is exocytosis. Let's begin with the definition here. It says here that exocytosis is the process by which cell move materials from within the cell into the extracellular fluid. Exocytosis occurs when a vesicle fuses with the plasma membrane, allowing its content to be released outside the cell. So what this basically means that exocytosis is the process of taking particle or substance from inside the cell, transferring it outside the cell using a vesicle. Now, this vesicle and cell membrane is actually made of the same substances. So when the vesicle touches the cell membrane, it infuses or merges with it and releases the content inside that vesicle into the extracellular fluid. Now, there are two types of process or pathway in exocytosis, and these are the regulated secretion and constitutive secretion. In regulated secretion, it says here that it is a process in which membrane of cytoplasm organelles fuses with the plasma membrane in response to a stimulation. This means that the vesicle inside the cell stays near the cell membrane and waits for a specific signal to trigger the secretion. Meanwhile, in constitutive secretion, it contrasts regulated secretion because it doesn't need or require any extracellular signal. In this process, components in the vesicle is secreted from the cell continuously. Again, the difference between regulated and constitutive secretion is that regulated secretion waits for a signal to trigger the secretion, and constitutive secretion, the vesicle is sent to the cell surface for immediate secretion.
Hello guys, I'm Kristel as Gravoso. As you can see, I'm here to discuss about the differences or to differentiate the endocytosis and exocytosis that been discussed by KARLO and ZYOMN. Let's look at the slide. Endocytosis involves uptake of substance from the outer environment, in which substance are brought into the cell, while exocytosis involves elimination of waste from the cell, where it removing toxics or waste product from the cell interior. Endocytosis helps to bring molecules inside the cell. It's also an active transport that moves particles such as molecules into the cell, while exocytosis releases enzymes, hormones, protein, and glucose to use in the other parts of the body. Exocytosis, the vesicle containing the waste substance fused with the plasma membrane, where the cell expels material through the fusion of vesicles with the plasma membrane and subsequent dumping of their content into the extracircular fluid, while the endocytosis involves formation of vesicle containing the substance. And that's the difference of endocytosis and exocytosis.
So I'm here again to summarize it in other words. So hypertonic solution has a higher concentration of solutes than the cell inside, or water will leave the cell and the cell will shrink. Isotonic environment is when both cell and the solution are of the same components, so no osmosis occurs, or there is no net water movement, so there is no change in the size of the cell. And hypotonic solution is a solution that has a lower concentration of solute to the cell. Water will enter the cell and the cell will swell. And in the endocytosis is a cellular process in which substances are brought into the cell. And exocytosis, opposite of endocytosis, is the process by which cell move material from within the cell into extracellular fluid. This process results discharge of material from vesicles at the cell surface to the outside of the cell. And that's all. Thank you for listening and watching.